Hello, and welcome to the 36th European Congress of Pathology. My name is Fayyaz Minhas, and I'm here to present my talk on Casualties of Causality. I'm sorry I could not be there in person due to visa issues, and I'm very grateful to the conference organizers first of all, for arranging such a nice conference, but also making special arrangements for my talk. The theme of my talk is to uncover some biases and confounders or in computational pathology deep learning models, or put more simply, ask the question, are we there yet? Can machine learning models replace machine learning models using just whole slide images of hematoxylin and eostin stained tissue replace conventional testing, for example, through IHC or through genomics and so on? I'm an associate professor of AI in biomedicine in the Tissue Image Analytics Center in the Department of Computer Science at the University of Warwick. Before moving on to the talk, let's take a philosophical look at what do pathologists do in the lab. So if this closest example I could find is Plato's allegory of the cave. So imagine yourself chained to a wall, unable to move and unable to look back at you. So this is you looking at some images that are cast in front of you by objects that you cannot see. So there are things out in the real world that we cannot see, and those are casting these shadows. And then our goal as these chained prisoners is to deduce what is actually going on out in the real world. This is quite similar to what happens in the pathology lab, in which we have got a disease mechanism, let's say giving rise to a tumor, but we are not directly able to observe it. What we do observe is the phenotypic effect of that disease in, let's say, hematoxylin and eosin stained tissue or radiomics or pathomics. And our goal is to use these um, phenotypic observations to make deductions about the pathological processes that may give rise to these. So in general, if you've got a disease mechanism, let's say giving rise to a tumor, then we can characterize it in terms of imaging from whole slide imaging or genomics or transcriptomics, or we can get immunohistochemical markers for that to deduce what is it actually going on in the disease, and thus what should we give to the patient to make them better. This is similar to an object casting multiple or different types of shadows that we can see, but we cannot see the actual object. And this is very beautifully represented in the title of one of my favorite books, Gödel, Escher, and Bach. You can have the same object casting different types of shadows. And all of these shadows, if you think about it, if you move the object, then the shadows should move across, all the different types of shadows should move across as well. And this concept is very beautifully illustrated in another recent book on how life works by Philip Ball. But relating it to pathology, back to pathology, is our goal as pathologists is to perform anti-causal prediction. That is, if we observe an effect, and then our goal becomes what is the underlying cause or the underlying pathological process giving observations of the phenotype. And we use that or we reduce that so that we can advise our oncologist friends better to make better decisions for the patient. We can use machine learning to help us in this case, in which we can take in these shadows or these images or genomic data or reports, and we can pass it through machine learning to act as a prism to gain diagnostic, prognostic, or predictive insights. So given this data, can we detect what type of cancer this patient has, or what would be the expected survival rate for this patient in the next 5 years, or what type of genomic patterns or biomarkers does this patient have, so we can give them appropriate drugs. So this is what my lab at the University of Warwick does. We use AI and ML to help discover and understand biology and pathology for precision biomedicine. Now, one of the in terms of computational pathology workflows, we know that we acquire a tissue, we prepare a slide, we can stain it with different inks. Hematoxin and eosin give rise to the most commonly or routinely used ones, and then we can either look down through a microscope, or if we have a digital lab, we can get a high resolution image, a very large 100,000 by 100,000 pixel image of that tissue, and then we can make machine learning models to help us diagnose different type of patterns or detect different types of patterns in the image to make decisions about the patient. So if we have a large data set of patients in which for which we have these images, and we have, let's say, what was their disease type, whether it was lung adenocarcinoma or lung squamous cell carcinoma, or whether in case of breast cancer, whether someone's breast cancer was HER2 positive or negative, then we can train a machine learning model by giving it the image as input and asking it to predict what would be the status of the receptor, in this case, in my example, HER2, for that patient. If the machine learning model's prediction is correct, we take it. Otherwise, we adjust the internal parameters of the machine learning model until it produces good results over the training set. And once it does, we say that the model has now been trained and we can now put it in testing. Our expectation is that the model should be able to predict correctly not only the examples of the images that it has seen in training, but also generalize to unseen cases, let's say from a different lab or from a different patient or from a different country altogether. So once that goal is achieved, we can then use just the machine learning model and replace conventional testing of receptor status with this image-based whole slide image machine learning model, which would help us get more efficient and hopefully solve other problems that we currently don't solve with imaging. So if can we solve MSI prediction with just imaging, or do we really need those MSI tests for this purpose? That is a question we are going to investigate. We all know that there has been a big surge of papers and methods in computational pathology, and now we are seeing some of those translate into the clinic with FDA approvals as well. But can we ask ourselves the question, are we there yet? But before that, we ask ourselves, what type of problems can we solve in using machine learning in pathology? So if we give you this image, can we ask ourselves the question whether the image, the tissue image that we see here, does that show someone, does it have any abnormality? Does the patient have a certain type of cancer, for example, or not? If so, what is the grade of that cancer? What part of these images constitute tumor regions? What type of cells are present? Or what is the receptor status of this patient? For example, whether they are HER2 positive or negative. What is the gene expression level of different genes in this for this patient just from the whole slide image? Remember, in machine learning, to train a method, all we need is pairs of images, images together with ground truth data. So if you got an image like this and we've got the expression level of this image for a certain gene, then we can use this pair, this labeled information, to train a machine learning model to predict what would be the status of gene expression for an unseen image. Can we also predict mutation statuses? Can we predict survival statuses? Can we predict the drug response? Can we retrieve similar images, or can we predict other type of biomarkers or identify different types of patterns in the image? So all of these problems occur on what my friend Nasir calls the spectrum of mundanity, in which you've got similar simple tasks of IHC scoring for which machine learning models can be used, to things that we typically don't solve with images, for example, MSI status prediction or HPV status prediction or mutation status prediction. So if we are able to achieve this end of the spectra, then can we really replace those tests? And how far along are we to replacing those? Or in other words, can we predict biomics-based biomarkers from whole slide images, from routine whole slide images using AI or using machine learning? I will be using both of the terms interchangeably, so don't get confused by that. And of course, the answer to that question is yes, because people have already published papers reporting this. So there is a very good body of work coming out from the community, very good high quality papers showing that it is indeed possible to just use whole slide images to predict, let's say, MSI status using just H&E images, or the BRAF or TP53 mutation statuses and mutations in other genes, or even gene expression levels. So this is quite well established that we can do this, but the extent to which we should trust this or the outcome of these methods is still in question. And that is the question we are going to discuss in this talk. There's also a wave of foundation models in computational pathology where people have trained large multimodal models. Some of the papers, very good quality work coming out from very good labs shown on this slide. For example, we have got this method called Virtuo, which essentially predicts the status of different biomarkers in various different types of cancer. For example, breast cancer, CDH1 mutations, or colon cancer, MSI statuses, and so on, with very high AUROCs that you can see over here. So, can we do pan-cancer biomarker prediction? There's already established work that shows that it is possible, but to be able to explain or to detect where we are or how far along are we built a machine learning model ourselves for pan-cancer biomarker prediction, so we could really do experiments on that to establish what biases and confounders affect that model. So for this purpose, the approach that we took is we take a whole slide image, we chop it up into small patches, and then we make a graph out of those. So each image is represented, because these images are very large, you cannot fit these images into the memory of a single GPU. So what we do is we chop up these images into different regions, and we characterize each region using a set of features. Those features can come in from deep learning models or other deep learning models, or they can be very pathologist driven. For example, they can be glands in a colorectal cancer case, or a colorectal tissue, and we can characterize, for example, what is the size of the gland, or how big it is, or how irregular it is, and so on. So each node in the graph is essentially a characterization of what is it at that particular location, and then the edges of this graph or between these nodes tell us where, or relative to other nodes, where does that part occur. Once we have this representation of the whole slide image, we pass it, or this graph representation of the whole slide image, we pass it to a graph neural network, and then we use machine learning training approaches to predict the status of different biomarkers like MSI, BRAF, TP53, or KRAS. We were able to do this using TIA toolbox, and the references to the toolbox, as well as the method that we have used that takes in a whole slide image and then predicts the mutation status or the gene expression status or even survival at the same time, just from that image, is given over here. So at the bottom, if you want to read more about it, I will not go into the technical details of how this method works, and you don't need to really understand how this method works in order to understand the next part, because this is not the only method we used. We also compared with other methods. For example, one very widely used method in computational pathology is CLAM, and we were able to get roughly the same prediction result or prediction accuracies for various different biomarkers using both of these approaches, as we can see on this slide. Sorry. Over here. So we have got the predictions. So in this case, we are showing the results of the training of different methods for different prediction tasks, for example, for ER, PR, HER2 prediction, or the prediction of mutations in CDH1, TP53, and so on. Or in the TCGA breast cancer cohort, using various different methods such as CLAM, which is shown in dark red, using different types of features representations. So there are different type of feature representations available that can characterize a part of the tissue. Here we are using something called a C-transpath, and we also use ShuffleNet, both using with CLAM. You can use other type of feature representations as well. We just wanted to see if methods trained with if the methods that have been using very different principles in their training, for example CLAM and slide graph, which work very differently from one another, to what extent are they able to predict pan-cancer biomarkers using various different feature representations, and how is the performance of these methods confounded by other variables that we will talk about later. But what we see, the good thing we see here, is that we are able to predict ER, PR, and HER2 statuses in breast cancer pretty well. Similarly, for CDH1 mutations or for TP53 mutations, the method does pretty well, and we are also able to get pretty good results on independent validation sets such as the CPTAC set or the ABCTB Australian breast cancer dataset for ER, PR, and HER2 prediction. So this is really looking up. That means that we don't need to, it seems like we don't need to do ER, PR, HER2 prediction if we allow a certain amount of error, of course, right? And we were also able to get similar results over colorectal cancer cases for various for various biomarkers such as MSI or mutations in BRAF and other genes that you can see over here using these methods. So things are beginning to look up. That is, we are able to predict these biomarkers using just routine whole slide images with machine learning models quite accurately. So now we ask ourselves the question, can AI replace existing tests using only whole slide images? To answer this question, we ask a series of additional questions. So, and if the answer to all of these questions is yes, then probably we can replace these tests using just machine learning or artificial intelligence. The first question is, are these machine learning models robust to domain shift? The second question is, are these methods able to predict individual biomarkers independent of other biomarkers or confounding effects? And are these methods really better than pathologists, or how do they compare to pathologists? Let's ask the first question. Are these models robust? In order to test this, we designed a very simple experiment, and you can read more about that in the paper that is mentioned over here. Now you see it, now you don't. What we did in this paper is we trained a very simple machine learning method to predict whether it could detect a tumor region or not by seeing at least one tumor cell in that. And the method was able to give us very good result with over 99% AUC ROC. So it was pretty accurate at detecting what parts of the image are tumor. So if, for example, for this image over here, let me switch to my laser pointer. Well, for this image, it was able to generate a correct prediction that this image does contain a tumor cell, and it was 97.6% sure that it does. However, we will not be able to see a difference between this image and this image, but there are subvisual variations or perturbations introduced into this image, which make the same machine learning model over a very similar looking image say that there are no tumor cells in this image, which is of course wrong in this case. And the point of this exercise was to show that machine learning models can be quite fragile. So they may be fragile to these perturbations, which may be subvisual, and we should really take care when training these methods to ensure that these methods are robust. To help the community with this, we built a toolbox called REAT, Robustness Evaluation and Enhancement Toolbox for Computational Pathology, which gives away different methods of assessing how fragile your method is, your machine learning model is, and how can you train it better to be less fragile so that it is able to perform better if it sees images that may look different or come from a different scanner or with different resolutions or even with different compression ratios and so on. So if you are interested in that work, please do explore that. But the conclusion is, unless we train these methods to be robust, they are not robust by themselves to such variations. With the development of foundation models, I must say that this, due to unsupervised training, the effect of such robustness issues has diminished, but we still need to test how robust these machine learning methods are to such variations. Similarly, when we have images coming in from different sites, let's say our goal is to predict p53 mutations in breast cancer cases or ER status prediction, then if we train our method on the data from a certain hospital, and then we expect that the method should generalize to other hospitals as well, but does it really do that? Now, one big source of variation between different hospitals or different sites is their staining, which you can see over here. These four images are taken from, these four images are taken from 4 different sites. And as you can see that unless we apply certain stain normalization procedures, these images just look very different from one another. But once we do apply the stain normalization procedure, they kind of look roughly similar in terms of their characteristics. And stain normalization has been used in the community to make sure that your models are not sensitive to such variations and are able to perform well or generalize well to unseen cases from other centers. But what we show in our paper, which is actually titled as "Do Tissue Source Sites Leave Identifiable Signatures in Whole Slide Images Beyond Staining?" What we show is that even after stain normalization, the information about the site is not entirely taken out, because we are able to design a machine learning model that can predict what source site or what hospital did the image come from even after it has been staying normalized. Thus, our conclusion is that machine learning models by default can gravitate towards predicting or confounding themselves with the site information, and we need robust testing in order to overcome this. So if you are interested in reading more about it, the link to the paper is at the bottom of the slide. Now, the more interesting question is, are models sensitive to other confounders? And the answer is that if a model should be able to predict the biomarkers in status independently of other covariates. So for example, let me switch on to here. Let's say if we design a machine learning model to predict the TP53 mutation status, then the only thing that that predictor should base its prediction on is mutations in the TP53 mutation, in the TP53 gene, and it should not be confounded by, let's say, source site. So its performance for TP53 mutation prediction across different sites should roughly be the same. But more importantly, its prediction, the prediction of the model, should also not be confounded by variables such as grade or mutations in other genes. Or in other words, if the prediction of a machine learning model is confounded by the effects of these different of other confounding factors, we need to be very careful because the model is then not predicting the individual effect of the variable we were interested in, but the combined effect of multiple markers. So do machine learning models actually predict what we think they predict is the question that we're asking here. We answered this question using something called stratification analysis and permutation testing, and I welcome you to read it, but the principle is that if we have a machine learning model, let's say for TP53 prediction, then it's if the prediction of that model, if the prediction of TP53 mutations by that model is independent of, let's say, grade, then if I stratify the patients in the test set with respect to their grades, the performance of the model across different grades should be roughly be the same in comparison to the performance across the whole cohort. And if that's not the case, then we have got confounding factors. If you want to read more about it, look at Simpson's paradox on Wikipedia, and that's a great read to explain it further. We developed, in order to test this, we trained a machine learning model to predict the mutation status of TP53 in colorectal in breast cancer, and it gave us this close to the state-of-the-art performance of 0.8 AUROC. But then what we did is that once we stratify the performance of the test set with respect to grades, then the expected performance of grade 1 patients should be around 0.8 or 0.77. The expected performance for grade 2 should be around the same, 0.77. And the expected performance of the for grade 3 patients should also be the same if the model is basing its prediction solely on TP53 mutations and not grade. But what we observe is that the observed AUROC of the model for grade 1 patients is 0.65 and not 0.77, and this is a statistically significant result. So the model's prediction accuracy goes down when we stratify with respect to each grade, which is really bad in the sense that our model is not predicting the effect of mutations alone, but it is predicting the combined effect of mutations in the TP53 gene together with grade. We also observed similar confounding effects in other genes as well. So this is a visualization of the results of the same model shown differently. On the x-axis is stratification by histological grade, and TP53 is the variable we are interested in predicting. And over here we see that our model's predicted performance across the whole cohort should be around 0.8. But once we stratify it with respect to grade 1, 2, or 3, the performance significantly drops, thus showing that the biomarker status is confounded by grade. And it's not just TP53 mutations, but it is also HER2 prediction, PR status predictions, ER, CDH1 mutations, and the pattern holds itself when we test it in independent test sets as well. Moving on. So it's not just that the mutation status of TP53 is confounded by grade, it's also that the mutation status of the prediction status of TP53 is confounded by other biomarkers. So for example, if we take TP53 mutations and we divide the patients in which PR expression is high versus PR expression is low, then we see that there is a significant confounding effect. So the model does not perform as well as it does in the overall cohort in each of these two different subgroups. And also the same story for ER prediction and for CDH1 mutations and for other type of mutations and biomarkers as well. So the conclusion that we are drawing from this is that ML predictions or machine learning predictions of a biomarker are confounded by other biomarkers. Similarly, when we train a machine learning method to predict mutations in a certain gene, then we expect that it will not be affected by mutations in other genes. But what we found is that if we, for example, train a method to predict the mutations in TP53, then if we stratify the patients into patients that have high number of mutations in other genes or low number of mutations in other genes, then the model performance is actually confounded by mutations in other genes, which will not happen in a genomic test. So this is not nice. The same story holds itself in colorectal cancer. I'll skip that over here, but MSI prediction can be confounded by, for example, chromosomal instability or hypermethylation statuses and so on, and similarly the same story for different types of mutations. So the conclusion that we draw from this is that AI predictors predict the combined effect of multiple biomarkers and are not able to predict the effect of individual biomarkers independently. So why would that be happening? And the reason for that is that in the training dataset, the covariates that we have, for example, grade or mutations in different genes, they are codependent upon one another, as you can expect. So tumor mutation burden, for example, affects grade, HER2 status affects grade, ER/PR status predictions indirectly affect grade as well, or are dependent upon mutations in other genes. So it's not just one gene that is, or the mutation in one gene that is causing everything. So isolating or disentangling these effects in training data is really hard. And why this is a problem is kind of like this. If we train a machine learning method to predict, to train it to detect cows, and the only examples that we give it are cows in grasslands like these, but then in testing, if we give it cows on a beach, it may fail. So as long as those correlation structures or codependency structures are consistent, it's not a problem. But in machine learning or in computational pathology, depending upon what data sets you pick, those correlation structures may be different. And we found that if we take, for example, the colorectal cancer, the TCGA breast cancer cohort and the metabric cohort, then the correlations between or the associations between these different genes can be quite different. And this may be one reason of why machine learning methods may fail if they've been trained on one data set and then tested on another. And we need methods to handle this. It's the same story for colorectal cancer cases. And lastly, we asked ourselves, are machine learning methods really better than pathologists at predicting, let's say, mutation statuses? Pathologists don't predict mutation statuses directly from whole slide images, but they do assign grades. So can we use grade information as a proxy to predict mutation statuses? And we found that yes, because of this correlation structure, you can predict, pathologists can, just looking at ER prediction and assigning a grade, they can predict the ER status as well as TP53 mutations to a sufficiently high accuracy. For example, for TP53, their grade information assigned by the pathologist in UCEC can predict the AUR predict TP53 mutations with an AURC of 0.8. For the CP-TAC, they can predict it with very high accuracy of exceeding 0.8 at times as well. So is there a solution to this? First of all, we need to make sure that our methods, so what we have established so far is that machine learning models may not be able to predict what we think they are predicting. And one way of overcoming that is to ensure that we have very large number of testing cohorts, so we do very good testing. And we also make machine learning models better by improving their transparency as well as making group predictions. And one way of handling that is given in the paper at the bottom. So if you're interested in reading more about it, you can read that. All of this is summarized in our latest preprint. which is available to you on this. So if you want to read more about these confounding factors and biases, you are welcome to this. So the conclusion is, can AI replace other tests? So are the methods robust? Unless appropriately trained, they may not be robust. Are they able to predict individual biomarkers independent of other biomarkers or confounding effects? The answer is no. And machine learning models may not be predicting what we think they are, so we need to be very careful. And are methods better at predicting omics biomarkers than pathologists? Most of the time, yes, but pathologists aren't that bad themselves. Just that we haven't figured out a way of training them to predict biomarkers directly from host slide images, routine host slide images. The solution to this is to actually model causality in machine learning model design to overcome these limitations and confounding effects. And this will not just be done by increasing the amount of data. However, we do need more data. So if you're a pathologist working with a computer scientist, I welcome you to give them more data with labels and your insights for testing as well as model design. I'm very grateful for all of this, for the excellent team that we have, and I'm especially to Daud, who did a lot of these experiments that I talked about. He recently graduated as a PhD student in my lab, and is now doing a postdoc at Oxford. I'm very grateful again to the community, and I look forward to hearing your questions and comments on this work. Thank you.